Welcome to the Mucosal Immunology podcast, brought to you by the Society for Mucosal Immunology and Mucosal Immunology Journal. Each episode will feature in-depth interviews, discussions on the latest scientific research, and real-world applications that are transforming healthcare today. Available on all major podcast platforms.
Marian here with Dr. Ashu Mangalam for a reoccurring segment of Mystery of Medical History, where we lead you through a mysterious mucosal disease. Ashu will provide a clue every episode for what disease he is describing. Hi, Ashu.
Hi, Marianne. Thanks for the introduction. And as you may have said, I am Ashu Mangalam, a longtime SMI member, and I am at University of Iowa, Iowa City, USA. And my lab is commonly known as Poop Lab because we work with gut microbiome and immune system in multiple sclerosis.
Poop Lab, I will definitely remember the name of your lab and what you do, Ashu. So this is a great addition to the Mucosal Immunology podcast, this little puzzle. How did you get the idea?
Actually, I'm very fan of a radio show called Car Talk, where two brothers, they're known as Click and Clack brothers, they talk about car problem. But what was more interesting is that in every episode, they have a puzzle where a puzzle man will come and talk about some puzzle where readers will send in their answer. So that's where I got this idea from.
Excellent. I'm sure it will be very interesting to our viewers. So the puzzle of today is called The Mystery in the Streets. So we are now in 1854 London.
So cities in this era, which is Industrial Revolution, around that time, so despite their advancement, were often vulnerable to outbreak of diseases because sanitation was poor and understanding of disease transmission was limited.
Yes, that's right. People were falling gravely ill and very fast, but amidst this chaos, a lone figure emerges, right? And he is determined to uncover the truth. So we'll call him John Doe for now, because his true identity is part of the puzzle. This John Doe meticulously documents each case, marking the locations on a map of London.
And this methodical approach to disease investigation was quite novel for the time. It foreshadowed the development of epidemiology, the study of disease patterns and causes. So, listener, it is your turn to join the investigation with me. Can you solve this two-part medical mystery?
So if you have an idea, please email your answer to mucimmunol@gmail.com. That's mucimmunol@gmail.com with the subject line podcast clue.
So here are three clues. So can you identify the disease plaguing London in this mystery? The clue number one is a nearby brewery, despite being in the affected area, reports no cases of the illness amongst its workers. They have their own private source of certain daily necessity.
Clue number two, a woman living miles away who has a specific item delivered to her home daily from the affected area falls victim to the disease along with her niece.
And the clue number three, areas of the city where a particular service relies on resources from downstream of the city's sewage outflow have a significantly higher rate of the illness.
Now for the second part of the puzzle. Who is this mysterious John Doe who bravely seeks to uncover the truth?
Our John Doe shares his name with a character known for his wisdom and sense of justice in a popular fantasy series on HBO.
One with dragons, right?
Yes, you're right, Marian. This character also faces a struggle against devastating force threatening his world.
Okay, that's quite a good clue, so it must be easy now, right? Send us your answers. The first listener to solve this historical medical puzzle, identifying both the disease and the mystery hero, wins a shout-out on our next episode and a place in our Mystery of Medical History Hall of Fame. Please email your answers to mucimmunol@gmail.com. That's mucimmunol@gmail.com with the subject line podcast clue. Thank you so much, Ashu. See you next time.
See you next time.
Now, for our Mucosal Immunology Research Roundup, we bring you highlighted research published in the latest issue of Mucosal Immunology. So sit back, relax, and choose what to read next. Check out the latest issue of Mucosal Immunology online now at mucosalimmunology.org. This issue includes a literature review from the Itch Dr. Brian Kim with John Mannion and colleagues at Mount Sinai on how epithelial barriers interact with the sensory nervous system. Mucosal neuroimmunology, or the study of neuroimmune interactions at mucosal surfaces, is an emerging field. This review covers the two complex systems of immunology and neuroscience, and how their interactions in the skin, lung, and gut inform immunity. Brush up on your neuroimmunology and how neurons respond to pathogenic threats with this in-depth review. You'll find the latest data in the research articles ranging from ulcerative colitis to allergic airway diseases. Here's 3 bites to get you started. First, Mahima Swami and colleagues at the University of Dundee in Scotland find distinct cell death pathways induced by granzymes collectively protect against intestinal salmonella infection. The authors describe how granzyme B can induce apoptosis, while granzyme A and B combine to induce a distinct type of cell death, paroptosis. Read more to find out how these two cell death pathways can control salmonella infections. Next, Lingli Dong-A, Jixing Zhong, and colleagues at Huzhong University in Wuhan, China, explore treatments based on myeloid-specific JAK2 inhibition. The article, Deciphering the Therapeutic Potential of Myeloid-Specific JAK2 Inhibition in Acute Respiratory Distress Syndrome. It uses mice deficient in JAK2 and a model of bacteria-induced acute respiratory distress syndrome to understand how inhibiting this pathway in monocyte-derived alveolar macrophages can reduce pulmonary inflammation. Finally, Crystal Loving and colleagues with the United States Department of Agriculture in Iowa use spatial transcriptomics to map B cells dynamics in the pig intestine. In the article titled Conserved B cell signaling, activation, and differentiation in porcine, jejunal, and ileal Bayes patches, despite distinct immune landscapes, they find similar B cell activity throughout the length of the intestine. Read more and enjoy the stunning spatial transcriptomics maps that are featured on the cover of the issue.
You can read all this and more at mucosalimmunology.org, home of the Mucosal Immunology Journal, the SMI-sponsored journal dedicated to all things mucosal. You can find reviews, primary research, and research collections from our entire archive, all available open access. Are you ready to submit your research to Mucosal Immunology? Your format-free initial submission will be given a first decision by an editor in less than 1 week. If you have a shorter story to tell, try our brief reports with up to 4 figures and a combined results and discussion section. Submit today on editorialmanager.com/MUCIMM.
Grab your lab coat or your driving wheel or a cup of coffee. I am Marion Brunk, and with me is Agnes Garcia, your host on the Mucosal Immunology podcast. So today with us is Dr. Régis Joulia. Hi, Régis.
Hello.
Very nice to have you here. You're an SMI member and you're also helping with the membership committee. Thank you for your time this morning. Could you please introduce yourself briefly? Let us know where you're working, what you're working on, etc.
Yes, hello. So thank you for having me today. I mean, it's a really great pleasure to be invited to speak to the Mucosal Immunology podcast. So, yes, I'm Régis Joulia. I'm working at Imperial College in London at the National Heart and Lung Institute. And I recently officially started my group of research at this institute and we are mainly working on the lung mucosal immunology, and we are doing analysis basically of how respiratory diseases are impacting lung vascular function, looking at how basically immune cells are interacting with blood vessels and how they can shape each other during development, and how mainly asthma for now, as asthma is shaping a bit and reshaping this immune response locally, and that could drive some of these changes that we see in patients. And we use both mouse model and human samples, and we used a lot, because I did this a lot in my previous background, a lot of imaging method, because we think it's super important, and to just analyze a bit more how spatially cells are coordinating with each other and how they can speak with each other, which I think is something that is very now trendy, but we did this for quite a few years, and we love it. I think it's really important to start better understanding what's happening in 3 dimension.
Yes, so that's very interesting, Régis. I think I can hear an accent. Did you study in France? I assume you were born in France, but when did you move to the UK?
Yeah. I mean, it's quite obvious from my accent. I mean, I do like to keep it. I think it's still like definitely something that like I will never be able to completely remove from when I'm speaking.
I think it's beautiful with accent. You should
Thank you, Ernest. Yes, and you can also pair around with the French, like, yeah, I didn't understand, I'm French. So I still use it a lot. So yes, I did my PhD and most of my studies in Toulouse, which is a small, relatively small city in the southwest of France. I did all my study there, so my undergraduate, master, and PhD there. And this is where I started basically really to be passionate about immunology. And my PhD supervisor was really, really great mentor. He just helped me a lot to shape some of these ideas together and teach me a lot about immunology, but also how to be curious and in science in general. And following my end of my PhD in 2016, I moved to London at the beginning of 2017. That was in the East London, and it was a Queen Mary University. I was with Professor Susan Nursha, and we did basically I changed a bit. My PhD was focused on human immunology, and I wanted to learn a bit more about mouse and in vivo work. So I went to her lab because they were specialized in intravital microscopy, and they were doing a lot of neutrophil blood vessel interaction. And I had always an interest in mast cell and blood vessel because they are closely associated with each other. So it was really a question of like merging of both our expertise in both my mast cell background and learning from the intravital. And I did a lot of mouse work, in vivo work, and stayed there for a while for the postdoc. Then I wanted to maybe go back to France, and then COVID happened. I stayed longer for the postdoc. And at the end of basically, I think the end of COVID time, Imperial College was interested to recruit me. We had a very nice chat, and they offered me some kind of transition step fellowship, where I basically have enough money to start running a lab, and after getting basically a stepping stone where I could get after maybe a more lab base. And I moved to Imperial, and after I was like, well, let's try it. If I get a lab there, it's great. If not, I can still move back to France. And
here we are.
yeah, and here we are. So yeah, they actually managed to convince me to stay, and I got the grant to start my lab. And so yeah, after this now I decided to stay here because I was quite happy.
Well, that's actually a very nice story, and I wish to say congratulations on starting your own group. Is quite an achievement these days, right? And I wonder why they wanted to hire you after a pandemic on a respiratory virus, right? And working on respiratory immunology and on lung immunology very much makes so much sense. So going to the research. Very interesting topic, very interesting techniques with all the imaging. I think it's quite impressive. So you actually published a research paper earlier this year in JCI describing a specific role for mast cell activation in early life allergic asthma. So what would you say is the most relevant finding of this work?
So yeah, that was no, I think it was. I was really happy. So personally, on this paper, I was just super proud because it was my first corresponding author paper, and doing this work in parallel of writing grant to set up a lab was just like a massive amount of work. And at the time, I was like, yeah, just continuing whatever happened. And actually, we found out it was really interesting thing. So the lab when I arrived, but my department, I would say, is interested into early life immune responses because this is really an area that is completely understudied, we think. And a lot of kids have very serious respiratory diseases. I mean, asthma is like impacting about 10% of child, 13% of child. And the immune response taking place during early life are completely different, completely regulated by other mechanism. And they started to do this to some extent, and we showed that there was learned very different immune responses, and this was a work of Sejal Saglani and also in the lab showing really like how they can differ and how T cell response are different. But when I started really, we wanted to know if the vasculature was different in this animal because early life is a period of development for the lungs at every basic component of the lung is developing, including the vasculature. But how asthma and how allergen and chronic inflammation were disrupting this vasculature was completely unknown. So we really just literally was just a characterization of how if you expose mice to allergen during this critical period of time, how the vasculature is changing. And the main finding that we found is that there are massive, massive changes in the vasculature that are related to not an actual, I would say, more blood vessels, but a loss of blood vessel and a very dynamic response. So the tissue can really adapt itself to how they're responding to allergen. And this was mediated, we believe, through parasites. Parasite basically changes in adaptation. So parasite, if you are not very familiar with them in terms of immunological response, that's perfectly normal because they are just very highly specialized vascular cell type. And but again, we know nothing about immune response. And the main finding that we found during this paper is that these parasites are actually very susceptible and basically very dependent of immune response, and they can really respond and be degraded and remodeled by the local immune response. And we believe that was partially mediated through mast cell activation, but not only. So I would say that if I had the main finding to say from this paper is like lung vasculature is super important and immune responses during early life are very different. And what we try to now understand a bit more is how a tissue that is still under development can deal with chronic inflammation and how this can have lifelong defects. And that's completely unknown also as well. So we believe that this is important, and we are trying now to explore a bit more the mechanism of how this is working in a bit more fine detail, because this was really just, I would say, one of the first stepping stone for my lab, what is this paper, and we are now exploring different aspects on how we're going to basically improve and better understand vasculature and mast cell activation during early life.
Oh, that is very interesting. And I actually have a question regarding that because you are the very example of this interface of immunology plus, in this case, vasculature, right?
Yeah.
And also, we know how important the first years of life are regarding the immune system development, right? And you work on allergic asthma, and then my question is, what about non-allergic asthma? For example, there is clear evidence that early infection with RSV can lead to late asthma, and I wonder if you also have some kind of work on that, and if the vasculature, this eruption can also be an effect on the RSV, so the non-allergic. Do you work on that, or is another big melon?
So I'm not going to lie. This is about in 90% of the case I have this question. It's just like
Okay.
ASV infection comes always. So we actually did it at the end because I was curious at some point. I'm going to do it, and we did it, and it didn't work great because I think one issue we have with mouse model is they are too clean, and our mice, when basically they see an infection, they're just happy because they are just, oh, we're going to mount actually a normal immune response to a virus. And for them, it is actually, we found it, and we still try to understand better what's happening. But it's actually even more protective for some of these mice to have an RSV infection before allergen model because they will actually mount a normal immune response. And after, when they will see an allergen, they will be like, oh, yeah, that's not maybe a real threat comparing to a virus. So it's actually maybe not worth going for like such massive immune response as you see in children. So it's very difficult to model the RSV infection. We are still working on this. We are still trying to find the best like way to approach this question because that's super relevant for human responses and human asthma, especially in kids. But for now, we don't have a response. So I have no idea if virus really impacting mast cell, are they impacting blood vessel function in the lungs? Are they doing so? We generally don't know so far, but it's definitely something we are looking at and continuing to explore.
So, Regis, you're actually a mast cell expert, right? This was your initial expertise, from what I understood when you mentioned your training. So, and I learned about mast cells quite recently, but I've started being bombarded with it from students visiting the lab and telling me they are working on them, etc. So I started reading about it in preparation for this podcast. And the role of these cells in allergic asthma is going through a little boom. And there's a lot of work getting done right now from the role of these cells in promoting type 2 inflammations, airway remodeling, etc. Possible translations, right? How does this knowledge on mast cell may help find some treatments to allergic asthma? So what do you think are the crucial questions that need to be answered right now to promote the advancement of this field?
That's a very good question. I mean, it's something I've been asking myself since I started my PhD, really. Because if you ask me, as compared to all immune cell, what are the function of mast cell? Like a normal function. Don't give me like some kind of like pathological function. Now, I won't be able to answer you. There won't be any kind of like clear definition of what mast cell are doing in terms of like why do we have them from embryonic stage? Why are they present around blood vessels? And you'll find, they're protecting against bacteria or viral infection, but how they do this, which is different comparing to a macrophages, I have absolutely no idea. So I think the most important question will be just a simple one. Like if you remove mast cell, or if you just look at them in terms of like biological assay, what are they doing? Are they protecting, and how they do it? Is it their granular component an essential function? Why do we have them from birth? Why do we have them from embryonic stage? Are they responsible for some kind of key biological, just beyond even immune response, but just key biological action that we don't know? I've generally no idea. That's super. I think if it's just really fascinating thing because everything is open. Are they interacting with neurons? Are they interacting with epithelial cell? Are they interacting with fibroblasts to direct tissue remodeling and tissue development? We generally have no idea about what they are doing. And this comes from, like everything in science, it comes as wave, and I think mast cell wave completely decreased for a long time, and it was completely absent even for more immunological causes, except a few groups that maintained the field alive, like Stephen Galley and Soman Abraham and others that maintained this field alive. But there was not really any kind of broad interest around them because we no idea about what they are doing. If you say a macrophage is what has primary function, you say phagocytosis, it's great. A mast cell primary function, no idea. Like generally no idea. So I think if I manage to do this with my lab and saying like, you know what, like by the end of my career, if I manage to find the normal function of mast cells, saying this is what they're important for, and this is what you could put in terms of like a immunological like lesson saying this is where you have them, and this is why we're going to keep them during our evolution, and we still have them now. That's what would be a great question to answer.
Thank you so much, Regis, for these insights, and I look forward to interview you in 30 years so you answer these questions.
Yeah, me too.
I actually have a question with that, very simple one, and I'm sure it's a yes. Maybe you can tell us a little bit. Has any scientist done the classical model of in vivo removing mast cells, like depleting mast cells, and see what happens, both in homeostasis and upon infection or so, and what happens?
Yeah, I mean, yeah, as you said, like people have done it and quite a lot, but I think we go back to the question of clean mice. And if you remove mast cell, you deplete them, and there are like new really good model now to deplete this, nothing happens really. Like mice are
Yeah.
fine,
Yeah.
but these are still laboratory mice. And I
Yeah,
think
yeah.
something that I think we have this question. I think like the group of Rebecca Gentek in Edinburgh is really key leader on this. Is like, and that's true. She made the observation that if you look at all clinical data ever reported, there is not a single human deficiency for mast cell. There is not a single case ever reported of mast cell deficiency in human. So either they don't have any phenotype, which is not very likely, but maybe it is, which will be very sad for my field. But I mean, it will be like just a possibility. Or they are so important that we cannot get rid of them, and they are not existing. So are mice the good model to look at them? Maybe not. Maybe mice are good to look at some specific function of mast cell. We had some data. I remember starting my postdoc looking at these mast cell deficient mice and seeing that other immune cells try to compensate for them. So we had more. We never published this because it's not really. I mean, to prove this is really hard to just show that there are more macrophages and dendritic cell, and that's maybe some compensation there happening there. But really, what after this, we really don't know what they are doing.
Yeah. No. Okay. So actually, there is a lot of question marks and a need for more research in mast cell, definitely to understand the function, right? So in this case, I'm sure all the mast cell experts are in contact, or you follow each other on the research breakthroughs, right? So what other SMI member or colleagues' work do you follow closely for answers and to collaborate for answering these burning scientific questions on mast cells?
And so on mast cell, I'm going to be honest, I don't think actually like SMI member, I wouldn't think I see any SMI member working on mast cell. The closest will be Bart Lambrecht, working in Belgium, which is a leader in type 2 immune responses. They have done work recently on basophil or other closely related innate immune cell, and including mast cell and IL-33. And his work has been always someone that we followed very closely and looking at this sort of question. After in my department, I'm very lucky to work in a very strong lung immunology department, and I'm working closely with Professor Claire Lloyd and James Harker and Cecilia Johansson that are all basically in my institute. And we, even if they are not working directly on mast cell, we all have shared interest into how these innate immune cells are responding to lung inflammation and dealing with lung injury. So that's something that we're all working on. After, SMI member or colleague, I would love that more SMI member work on mast cell. Unfortunately, I don't think that there is so many working on them. So it's something I definitely try to push at the next meetings and congress. But yeah, for now, I think it's still a struggle.
Maybe this is a good shout out for all the immunologists and SMI members there that can work a little bit within the interface with mast cells. They can contact you.
Yes, definitely. Yeah. There is a lot of I think just there's enough people working on ILC and macrophages. I think we can like also start to work on other cell type. So that would be quite good.
So it sounds like you've had great mentors. So I know Suzanne because I'm a neutropenia person initially, and also you've participated in the mentorship program of the SMI, right? So I'm actually in the current cohort. So I wanted to ask you about your experience, please. Could you share how was it for you?
It was great. No, it was great experience, and so I started. In which cohort I was? I was in the cohort of 2022, and I was paired with Ivan Zanoni, which he is in Harvard Medical School, and he works also as well on. He works on a lot of things, but it's a lot of related to cytokine and interferon regulation of infection, and it's not directly related to my subject, which was good because it also brings also as well a different aspect. And he was also not UK based, which was good in terms of having completely an independent feedback. And I chose the really, I put several people, and I think he was one of the best one I wanted for, because he's also as well, we come from both an European background, which is also helped, and I'm from the south of France, he's from Italy, and we had quite a different, like, similar mentality, but which was just easy to sometimes discuss things comparing to more, I would say, Northern European type of culture. And so we had a chat after when we started this program. It was really good because he's also as well a very honest person. So if I came up with ideas saying, oh yeah, I thought about this for a grant application, or we have this sort of data, and he was very honest and straightforward, saying like, yeah, no, this is nonsense, like you will never get this funded, or you like, yeah, it won't, no, that won't be interesting, or like you definitely need to explain this more. So it was really good in terms of like, and I'm also a person that really appreciate honest feedback, so I prefer to just having someone telling me this is like, yeah, this is nonsense, and just move around, instead of just someone trying to be nice and like also which is a very British way to do it, but like I still prefer like the more I would say like French way, or so to do it. But I think it was a really great experience. No, I really enjoyed it. I went to visit him last year. He was also very nice. He arranged a seminar for me at Harvard Medical School. When you start your lab, this is really terrifying experience, because you have all these professors from department. It's just like, well, here we go. Let's start about my ideas. But that was brilliant. Really, really nice experience. I will highly recommend it. Because it's also at this stage of your career, which I think is really helpful to get some feedback in terms of to not go in the wrong direction, because you can easily at this point be by yourself and thinking, oh yeah, I'm going to do all of this project. And if you don't have someone who's just been through also as this stage and telling you, just no, restrict yourself to this one or two things, because if not, you're not going to achieve anything. But also simple stuff, recruitment, we discuss, personal life, family thing, traveling back to my home country, how many, is it easy? So it was really also a lot of simple discussion about like this period of life, and which was also as well not sometimes easy to discuss with primary old supervisor that are also at a different stage of their career. So that was also as well a super rich, super great experience.
Okay, and you had some some scheduled meetings? Did you have some scheduled meetings?
Yes, so we decided. I mean, it was a bit. He always made it clear from the beginning that it was me deciding the path and the schedule of these meetings. So how many times are we going to meet and what we are going to discuss? Because I was driving and applied for the scheme, and he was there and said like, I'm here to help you. I'm not really like here also as well to just force you to have meetings to discuss the weather if we just like have nothing to discuss. So yeah, we have scheduled meeting, and between this meeting we'll send email to each other saying, oh yeah, just apply to this. Do you have any like this is a quite quick summary? Do you have any feedback for me or these sort of things? And that works. But after each mentor-mentee relationship is different. I would strongly advise to try to find time as soon as possible and schedule an in-person visit also as well as soon as possible, because that's, I mean, the SMI is giving you money to do it, and not doing it is a mistake. So definitely go and make the most of it. And I mean, I spent a few days in Harvard. I met a lot of people, and the seminar made me even meet even more people after. So it was really a great experience.
How nice, Reg. It sounds very nice, a very nice program, and how important it is to have mentors in these moments of the career, right? When you are starting with your own group and how to do things. So overall, you would say the main advice on how to get the best out of this program as a mentee would be to be proactive and drive yourself the whole mentor-mentee relationship meetings, and so you can get the most out of it, right? What would be the best?
Yeah, I would say
right?
that
The proactivity.
Yeah, the proactivity, and you need to make sure that when you apply to this thing, you have something clear in mind that you want to, and you are not just hoping to just receive like information from someone. You really need to be like, me was like really like, I need to apply for a big grant to start my lab. I want feedback on if it's realistic or is it completely nonsense. And that's what I wanted for me. I mean, being at Harvard, it was very like, yeah, I mean, this is a tough place, and don't worry. I mean, like, if I tell you it's like nonsense, I'm going to like tell you straight away. So that was, I think, the best advice I had which was worked for me. But I mean, everybody has their own like after plan for at their stage of their career.
So I understand that during this time you applied to various grants. You were just mentioning that you applied to a few of them, and you're now supported by the Wellcome Trust, or you've been supported by the Wellcome Trust and major UK initiatives. Congratulations. So I guess you were receiving feedback from your mentor, possibly talking around in your lab to colleagues. You know, creativity is very important to get a grant accepted, funded. So I would like to ask you a little bit about creativity in your intellectual pursuit and grant writing. How do you foster it?
Yes. So yes, thank you. So Wellcome Trust now is funding our lab, and I mean, creativity is, yeah, when it quite annoys me when sometimes we have students saying, oh yeah, I choose science because I'm not a creative person. If not like creative person, do arts. And this is a big mistake, I think, because science is one of the most creative process that it's possible. If someone is not creative, I mean, you're just going to repeat idea of each other and not being original. And if you do this in science, this is not a path to where you're going to get fun at work, but also just manage to do and achieve anything. So foster creativity, it's really hard. I'm not going to lie, because how can you know like that something you're doing has not been done before? But what we try to do as a department is to that's something, I mean, Claire was always very keen on. Claire Lloyd, that was my previous supervisor, and what we try to maintain this as a department is to maintain the curiosity level as high as possible. And to do this, we try to really being like asking, not overthinking question, but asking simple question. So what will be the role of this cell in this model? Where this cell will be if you ask a simple question, where are mast cells located in the lungs? And if you're not able to answer into a simple term in a simple question, that means that the question needs to be addressed. And that remains creativity. So that's where we're trying to do this and push students and even ourselves all the time to ask a simple question. And that promotes creativity, I think, before the overthinking of the detailed mechanism of how it could work, but just asking like simple question that everybody could understand.
Yeah, this is a great tip, and I'd never thought of it this way. It's a great perspective. I'm gonna start doing this, Régis.
Yeah, no, I think it works. I mean, we tried it quite a few times, and Claire is doing it all the time because she just like, she just asked me simple question, and we had one recently where I was like, oh yeah, are mast cells moving between different locations of the organ? I couldn't answer to her. I really don't know. Maybe not. I don't think so, but I don't know. And if you're not able to answer it, and if you look quickly in the literature, you're not able to see a clear answer, maybe it's worth addressing then. And you go after down this step of like asking other simple question when you answer the first simple question. But yeah, we try to not, really important to not overthink sometimes the data and not overthink the experiment and after trying to answer, simple question.
That's so great. It's funny, the whole simple, not overthink, right? And that can bring you far. So now we move a little bit away from the science and to get to know you as a person. We have a little question of what's your favorite movie and why? Maybe you don't have a favorite movie. Maybe you can tell us a little bit of a hobby you have. You can choose yourself. Just tell us a little bit about you as a person.
Favorite movie? Oh yeah, I didn't. I was not expecting. I don't have a favorite movie. So I'm a big cinema fan. I watch a lot of movies, but I don't necessarily come from a favorite movie because I think like I like a lot of movie and they all have like really good movie for different reasons and depending of my mood, I like all Tarantino's movie. I think they are really quite brilliant and sometimes they are really nicely also as well done. And I really like the way he's filming also as well and building these movies, which I think are really great. I think we are also as well, it's really sad, but I'm also an imaging person. And I really like also as well how these things are filmed. I really like also as well after you realize how like filming this sort of thing and capturing this moment are really difficult. So that's why I think it's also a relationship between between my lab expertise and film. So I can relate. I'm just saying, oh yeah, getting this light like really according in this like landscape is really hard to see. So it just like must be a nightmare. So that's something. That's why I really like this sort of movies with like thing like this. After this hobby, I would say that it depends of the season and my mood. I do like, and this is something recent, but it's really sad because I used to. So my parents are very big gardener and they love their garden and they always did this. And as a teenager, I hated this. I thought it was just boring. I hated this and and that was, just waste of time. Why we do this? And since recently, as I get older and maybe I do like going back to gardening again. And as I move also as well out of the lab and my student and staff are just pushing me out of the lab because I'm not as efficient as I used to be. I do like gardening even more and more, which sounds boring, but it's actually true. I actually like to do this, but hobby is also as well a different thing. I mean, I recently, also as well bought a camera and starting to do some photography, which is also as well a very good hobby, and I love also to do this now. But it's also as well just an extension of my microscopy also as well skills, which is also no, I would say an hobby, but just basically another type of microscopy that I'm just using.
Yeah, well, it's great, right? That you can actually also use your skills
exactly
outside the lab. Think outside the box. Use it outside the box.
Yeah,
that's great. Well, Régis, that was it for us. You are welcome to have a last comment. If you have anything else you'd like to say.
Yeah, we have explored more or less quite a lot of things. I don't really like to speak too much about myself because I have a twin brother, and when you have a twin, you never like to be the center of attention because you're always like, well, two always. So yeah, no, it was a really great discussion. I really enjoyed it. I hope it's useful. I think it was really good, and thank you very much again for inviting me to speak today.
Thank you so much, Régis. It was awesome to get to know you kind of in person, get to know you personally, get to know more about Mast Cells. You made me want to read more about them. And I look forward to your next papers. Thank you for joining us.
Thank you.
Thank you to our content contributors, Drs. Marian Brunk, Agnes Garcias-Lopez, and Ashu Mangalam. This podcast is produced by Matt Close and Catherine Knoop. Finally, here's a sneak peek of a conversation between Drs. Peter Openshaw and Ryan Thwaites as part of the SMI's Oral History Project.
The emergence of your personal interest in this field and in the emergence of the SMI, do you feel like that was something coming together in the community at the time, an increasing interest in mucosal immunology?
Yes, I think there were a lot of people doing interesting stuff, particularly in relation to, B cell immunology of the intestinal mucosa, and it was such a crucial area for scientific development and collaboration. And I remember going to some of those early meetings and just being bowled over by the comparisons between what they were discovering in the gut and what we suspected might be happening in the lung. I think the lung was really lagging behind the gut. There were a lot of great immunologists who had started to try to sample and describe what was going on in the gut during mucosal responses, but relatively few people in the lung. So it seemed very important to go along and to try to emphasize the importance of the lung.
Be sure to check out the full-length version online now on SMI's website at www.socmucimm.org. We hope you enjoyed this episode. Remember, the journey of discovery in mucosal immunology doesn't end here. Stay connected with us for more in-depth discussion, expert interviews, and the latest research breakthrough. If you found this episode valuable, please subscribe, rate, and leave us a review on your favorite podcast platform. Your feedback helps us reach more researchers passionate about mucosal immunology. If there are questions or topics you'd like us to cover in future episodes, we'd love to hear from you. Connect with us on LinkedIn, Facebook, or Twitter/X. Thank you for listening.